species when they are reproductively active, or which prevent or restrict the union of gametes after mating or cross-pollination has occurred. It may be caused by the following reasons. 1. Habitat isolation, ecological isolation. Often two closely related species will thrive in different ecological conditions, habitats, within the same territory, but no hybrid between them will be found. However, when the members of the representative species are taken into the laboratory, hybrids between them can be obtained. Thus, their respective gene pools are isolated physically, but not physiologically. Again, because of the ecological separation that inhibits the sharing of traits, the incorporation of new adaptations may lead to functional separation of their gene pools, and speciation. Habitat or ecological isolation is most common in plants because of their sedentary nature. In them it operates in two ways. The species may live in the same general area but have such different habitat preferences that their populations are rarely close enough together to cross-fertilize each other frequently. In addition, if hybrids are occasionally formed, they may be either unable to grow maturity or leave few progeny under natural conditions, because no site to which they are adapted is available. This is well illustrated by several species of oaks. Many oaks are interfertile and hybrids are occasionally found. Such is the case with 4 Texas species: Quercus moriana, Q. havardi, Q. grissi and Q. stellate, studied by C. H. Muller, 1952. These 4 species are kept distinct because they grow in very distinct soil types, namely, limestone, sand, igneous outcrops and clay respectively, and the few hybrids between them are confined to the zones where the soil types come into contact. Furthermore, 2 plant species of scarlet oak, Quercus coccinea, and black oak, Q. velutina, are sympatric throughout most of the eastern United States and both are distinguishable by shapes of their leaves and acorns. Sympatry is the phenomenon of occurrence of 2 or more populations in the same area, more precisely, the existence of a population in breeding condition within the cruising range of individuals of another population. Such populations which are related and share a portion of their ecologic ranges but remain isolated from the another not by space but through the physiologic expression of genetic difference, are called sympatric populations or species. Habitat isolation is not a very effective isolating mechanism in mobile animals and has been observed most frequently in mouse, river fishes and certain water snakes. For example, 2 subspecies of American mouse, Paramyscus maniculatus, inhabit regions that share a common boundary. While laboratory hybrids between these subspecies exist, none has been found in nature. The explanation for this isolation is that one subspecies lives in the forests and the other on sandy beaches. Apparently, members of the 2 groups rarely encounter each other. The same situation has been found for another subspecies of mouse in Oregon as well as for the water snake Natrix sapedon. One subspecies of snake is a freshwater race, and the other is a saltwater race. 2. Seasonal olation, temporal isolation. 2 groups may exist in exactly the same ecological area, but do not interbreed because they become sexually mature at different times of the year or under different conditions. Further, seasonal barriers are particularly frequent among aquatic animals, because water temperatures are most stable than air temperatures and embryonic development is more closely harmonized to definite temperatures. These 2 factors combined and permit a close regulation of the breeding seasons. Seasonal isolation is common in plants and occurs frequently among insects and other INVERTE 118 evolutionary biology. Brates and some vertebrates. For example, over 10 subspecies of cypress trees, Cupressus, are found in California. They do not interbreed because each race produces pollen at a different time of the year. In animals it may be the time or condition for mating which may vary. In the northeastern United States, 3 species of frog, Rana pipiens, R. sylvatica and R. chlamydens all mate in the same ponds at different times. R. sylvatica begins breeding when the water is 44 degrees Fahrenheit and completes its mating season before the temperature reaches 55 degrees Fahrenheit, the breeding temperature necessary for R. pipiens. R. chlamydens must have temperatures of at least 60 degrees Fahrenheit by this time the temperature is reached. Both R. sylvatica and R. pipiens are past their breeding stages. Further, most closely related species of anorans, frogs and toads, have overlapping breeding seasons, but in them, the isolation is provided by acoustic stimuli. 3. Ethological or behavioral isolation, sexual selection. Ethological, GR, ethos equals habit, custom, refers to behavior pattern. Ethological isolating mechanisms are barriers to mating due to incompatibilities in behavior. It is based on the production and reception of stimuli by the sex partners. The males of every species have specific courtship behavior and females of the same species are receptive. Courtship involves an exchange of stimuli, visual, auditory, tactile, olfactory or chemical, between male and female continuing until both have reached a state of physiological readiness in which successful copulation can occur. The species-specific stimuli which help 2 sexes for the simultaneous recognition of each other for courtship forms the ethological isolation. For example, birds and insects embark on mating after highly standardized ritual dances. In some species of birds, plumage of the male stimulates the female's sexual interest. Thus, the different color patterns of insects, different color patterns of plumages of birds, light signals sent out by male fireflies, beetles of family Lampyridae, melodious songs produced by the males of insects, frogs, birds, etc., sex attractants, ectohormones or pheromones produced by most insects, mammals including human female, are some of the examples of stimuli which have a significant role in ethological isolation. In most animals it is the male that actively searches for a mate. He is usually rather easily stimulated to display to objects. Sometimes quite inappropriately, when he does not receive adequate response from his display partner, or is actively repulsed, his display drive eventually becomes exhausted. Consequently, If such a displaying male encounters an individual of a different species, or a male of Highzound species, he will break off his courtship sooner or later. If the male is displaying to a non-receptive female, the same will happen, perhaps, after longer intervals. In Drosophila, for example, it is shown that males act quite specifically to remove inhibitions, so that females will copulate with males of her own species. These actions of courtship include Orienta, wood warbler, chiff-chaff, quote, Cula, _F%. The English naturalist Gilbert White, 1720–1793, was the first to notice that the chiff-chaff, the willow warbler, and the wood warbler were three different species, and not just one. The wood warbler is slightly larger and brighter inch of the body, display of wings and licking with tongue. Further, when the antennae, the most sensy color, but the chiff-chaff and willow warbler look almost exactly the same. The songs of these three, however, are all distinctly different. For the birds, the songs are used by the female to select a mate, so in this way they act as an isolating mechanism, separating the otherwise similar species. Isolation 119. Type tactile organs of females are etherized. The males can easily bring about copulation with females of different species. 4. Mechanical solation. Mechanical isolation is provided due to differences in the structure of flowering plants or in the genital organs of different species in animals, so that cross-pollination or copulation may not be easily brought about. In case of higher plants, mechanical isolation provides the most effective and a major isolating mechanism. It is more effective in those plants where flower structures remain well specialized for insect pollination. For example, different species of Asclepias of Asclepiodaceae differ in the shape of pollinia, sacs carrying pollen, and that of the stigmatic slits, so that with the help of particular insect bringing about pollination, pollinia of one species cannot bring about pollination on the stigma of another species. Such kind of mechanical isolation is also common in the other members of family Asclepiodaceae and Orchidaceae. The orchid family has evolved flowers having an architecture that is particularly susceptible to large numbers of variations on a single adaptive theme. They are bilaterally symmetrical or zygomorphic, because one of the 6 perianth members are petals. Tepals is modified into an elaborate structure known as the lip, the santa, united to form a single I structure, the column, O, which in each species B1 produces a very specific A1L relationship between the Catalia flower P pollen-releasing anthers stigma, and their sticky pollen mass cannot be transported by any means other than a particular vector, figure 9.1. Unless the flower is visited by particularly adopted to Poi en Lum, RL section ECT1 or a few related species, no seed is formed. CGL Stebbins, 1_1976. Flowers of some O equals orchids even mimic in A3B3 shape the females of certain species of wasps and bees. Dodson, 1967. Pollinium B2. Mens and stigma are. And the pollen receiving. An insect or other visitor. Figure 9.1. Pollination of an orchid Catalia flower, a highly specialized and vector-specific flower. A-1. Face view of the flower showing the 3 narrower outer petals and 3 broader inner petals, the lowest of male wasps or bees or which is the lip. A2. L. S of the flower. Partly enclosed in the lip is the attracted to these flowers stamen that contains pollina, the stigma that receives pollen and engage in pseudocolum that support them. A3. L. S flowers showing position of copulation, with them, insect visitor in the act of delivering pollen and receiving anew thus, receiving in transpollinium. B. Enlarged sectional drawings showing how the faring the pollen sacs. Pollinium becomes stuck to the bee's thorax, after Dobzhansky, et al. Theocrins of 1976. 120 EVOLUTION Evolutionary Biology. Mechanical isolation among animals was recognized quite early. Soon after the discovery of the manifold structural differences in the genital armatures of different species of insects, it was asserted by Dufour, 1844, that these genital armatures act like lock and key, preventing hybridization between individuals of different species. For example, interspecific crosses in Drosophila may cause injury or even death to the participants, and the same is true in Glossina, tsetse fly, and in pulmonate snails of subfamily Polygyrinae. However, Carl Jordan, 1905 contradicted Dufour's hypothesis by showing that out of 698 species of Sphingidae family of Insecta, 48 were not different in their genitalia from other species of the family, while in about 50% of the species with geographic variation in color, there was geographic variation in the structure of the genitalic armatures. Since that time much additional information has accumulated indicated the slight importance of the genitalic armatures as isolating mechanisms in animals. According to Meyer, 1970, the genitalic apparatus of animals is a highly complicated structure and is the pleiotropic byproduct of very many genes of the species. Any change in the genetic constitution of the species may result in an incidental change in the structure of genitalia. As long as this does not interfere with the efficiency of fertilization, it will not be selected against. B. Postmating or POSTZYGOTIC isolating mechanisms. Postzygotic isolating mechanisms are those which prevent the growth of hybrid individuals after fertilization has occurred or which reduce the fertility of the hybrids or the viability of their descendants. They are caused by the following methods. 1. Gametic mortality. It has been postulated that sperm can tolerate only narrow ranges of physiological conditions, pH, temperature, etc., and that these conditions are favorably met in female members of the same species. Deviation from the physiological compatibility would prove lethal to sperm, and few, if any, would survive to fertilize the egg. Therefore, even though members of these two groups could copulate, the mating process would not lead to viable offspring. Further, the sperms may encounter an antigenic reaction in the genital tract of the female, so that they become immobilized and killed before they have a chance to reach the eggs. Patterson reported such insemination reaction to occur in many Drosophila, which leads to an enormous swelling of the walls of the vagina and the subsequent killing of the spermatozoa. In certain sterile human females, sperm mortality due to excessive acidic conditions of vagina has been reported by many medical journals. Sperm mortality may also occur due to physiological inability, i.e., fertilizin-antifertilizin reaction, of sperms to penetrate the egg membrane of the alien species. Such Gametian isolation is not unique to animals. In plants such as the jimsonweed, Datura, the sperm-bearing pollen tube of one species encounters a hostile environment in the flower tissue of the other species and is unable to reach the egg. See Volpe, 1985. 2. Zygotic mortality. The development of the fertilized hybrid egg is often irregular and development may cease at any stage between fertilization and adulthood. Zygotic mortality can be caused due to several cytological, genetical, embryological, molecular, biological, biochemical and physiological reasons in most animals. For example, certain hybrid zygotes of Ambystoma mexicanum lack nucleolus, and in the absence of nucleolus, such zygotes die without undergoing cleavage. Certain authors have included the isolations due to sperm mortality and zygotic mortality under physiological isolation. Such physiological isolation also can occur as an incompatibility between the embryo and the female parent. Interspecific hybrids between two species of jimsonweed, Datura, result in the death of the embryo at the 8-cell stage. By removing the embryos and culturing them artificially, viable seeds can be produced that germinate and grow into healthy offspring. 3. Hybrid inviability. Many naturally occurring animal hybrids though have somatic hybrid vigor and fertility, but isolation 121. Leave no offspring. The reason for the reproductive failure of fully fertile species hybrids is perhaps that they are less well adjusted to available ecological niches than individuals of the parental species. Also, species hybrids are usually less successful than individuals of pure species in courtship. When definite behavior place turns in species-specific stimuli play an important role. Ecological and ethological inferiority, thus, reduces their chances of leaving offspring. For example, two species of the chicory plant, Crepus tectorum and Crepus capillaris, can be crossed, but the hybrid seedlings die in early development. Crosses between the bullfrog, Rana catesbayana, and the green frog, Rana clamitans, results in inviable embryos. Rana clamitans. 4. Developmental hybrid sterility. Sometimes postzygotic isolation involves in the production of vigorous but sterile species hybrids. This type of hybrid sterility involves the abnormal development of gonads, or abnormal meiosis or abnormality in gamete formation. It is usually most common in male animals and plants, for instance, and viable hybrids between different species of Drosophila and flies, also between various species of mammals such as cattle x yak, cattle x buffalo, horse x zebra, and between some species of birds such as mallard and muscovy ducks. Thus, in certain hybrid crosses, such as between Bufo fowleri females of the toad species Bufo fowleri and males of Bufo valiceps, the hybrids may survive but are completely sterile. The abnormality occurs due to poor growth and a low rate of mitosis in the cells of seminiferous tubules. Further, developmental abnormality also occurs at the time of meiosis of spermatocytes, due to which either no sperm is produced or if any sperm is produced that remains indifferentiated, to be unable for fertilization. Muntzing 1930 has used the terms haplontic sterility, where the sterility is set in haploid parts gametes, and diplontic sterility, where the sterility is brought about due to events before meiosis or after fertilization in diploid tissues. Bufo valiceps. 5. Segregational hybrid sterility. In both plants and animals, hybrid sterility may result from abnormal segregation at meiosis of either whole chromosomes or blocks of genes contained in chromosomal segments. If the chromosomes of the parental species are not homologous, i.e., chromosomes of both parents of a hybrid contain individual gene dissimilarities, they cannot pair at all. Further, segregational hybrid sterility or genetic isolation may also be caused due to genetically controlled spindle abnormalities, a synapsis or desynapsis, and similar other abnormalities. For instance, in the hybrid Raphanobrassica between the radish and cabbage, the 9 chromosomes derived from the radish may not pair at all with the 9 chromosomes derived from the cabbage, so that at meiosis one observes 18 single chromosomes instead of 9 pairs found in the parental species. Since these unpaired chromosomes are unable to line up on the meiotic metaphase spindle, they are distributed irregularly to the poles cause daughter cells 122 EVOLUTION evolutionary biology. With unbalanced complements of chromosomes, the genes of which are unable to direct the development of the pollen grains or embryo sacs. Further, sometimes chromosomal sterility may result when the chromosomes from two parents do not lack homology but differ due to structural changes like translocation or inversions, chromosomal aberrations. In such a case, the pairing of chromosomes will be imperfect and results in the segregation to the gametes of abnormal disharmonious combinations of genes, i.e., sterile gametes. For instance, in hybrids in between Primula verticillata and P. floribunda, the sterility in hybrid is known due to small structural difference between parental chromosomes. Likewise, the functional offsprings, mules of horse and donkey, are sterile. The horse and donkey each contributes 33 chromosomes to the offspringer. Species hybrid mule. During meiosis of gametogenic cells of the mule, however, the chromosomes fail to pair. This failure is thought to be the result of the degree of genic dissimilarity between the horse and donkey chromosomes, so that they do not recognize one another as homologs. Since the orderly separation of chromosomes into gametes is necessary for fertilization to take place, the mule is sterile. There is evidence that genetic isolating mechanisms are occasionally modified or reinforced by conditioning. Young birds can sometimes be imprinted on a foster species when raised by foster parents. In parasitic birds, cuckoos and cowbirds, such conditioning is absent, and species recognition is rigid. Young cowbirds, for instance, leave the company of their foster parents and flock together as soon as they are independent. In the human species, however, conditioning is an important isolating mechanism. The free interbreeding of individuals coexisting in a geographical region is strongly influenced by religious, economic and cultural barriers. 6. F. Breakdown. In both animals and plants, there are examples of hybrids which are highly or at least partly fertile, but which give rise to weak, abnormal, or sterile progeny in the second F generation. This phenomenon is called hybrid breakdown. Classical examples of hybrid breakdown are provided by the F hybrids, Gossypium arboreum xg herbaceum and Gossypium barbadense xg hirsutum. In both of these cases, F progeny is either missing or is very weak. Primula verticillata. Primula floribunda. From the developmental point of view, F breakdown is similar to segregational sterility, except that the effects of the segregation of disharmonious gene combinations are delayed until after fertilization.